Thank you. So I am going to try. So hi, my name is Aleksandra Walczak. I'm supposed to give 3 lectures on the immune system. Okay, so we'll see what happens. Uh, I have 3 lectures. I don't have to get through with them. Does anybody know anything about the immune system? Yes. Okay. I'm glad people from my group are not raising their hands. All right, so specifically we're going to talk about adaptive immunity. So adapt, and basically today I'm going to try and sort of talk about the diversity of the adaptive immune system. So first of all, and specifically immune repertoire. So first of all, I'm going to try and explain what is an immune repertoire. And basically diversity, I'm going to try to say how big it is. Now, I'm going to, yeah.
Sorry that I make a comment that Massimiliano
Yeah.
should do in 5 seconds. So for the recording, so you see this line there.
Ah,
So
okay.
everything left right of it won't be on the screen.
Okay. I'm not going to say anything very important in the next 5 minutes. I'm going to say something very important for French people. I write my R's like this or like that. I don't do this. Okay? This is an R. Try to integrate that. I am incapable of doing this. I know it perturbs you. I'm sorry. All right. So immune repertoires, you have to, well, basically adaptive immunity. Okay, and I'll add the line here. Through a T cell or B cell. So I'm just going to write what I say. So basically for now it's okay. It's not super important. Adaptive immunity is made out of B and T cells. Okay, these are 2 types of cells. You may have heard of them. B cells are what you get when you go to get a blood test and they have a lymphocyte line. Those are B cells. Okay, T cells are similar. Anyway, you know about B cells and T cells because we had a COVID epidemic, right? Okay, good. All right, so that's what we're talking about. The way they look is like this. They're cells and they have receptors on the surface. And what these receptors do is they bind other cells, bits of other cells or bits of peptides, and they're able to recognize whether this cell or bit of cell is foreign to your body, in which case likely a pathogen, or whether it's natural to your body, which case leave it alone, right? So basically these receptors on the surface of these cells perform a discrimination task. Okay, break the chalk. Okay? They're able to recognize something that is your own protein, which we'll call self, versus something that is not your own protein, which we'll call non-self. And non-self means likely a pathogen. And they do this by binding molecules to receptors. See, there's the R, another R. Okay? Now, what's specific about them, and when they bind them and they recognize that it's a non-self, that means what they'll do is they'll launch an immune response. An immune response is the, okay, response. I'm gonna go down here. An immune response is everything from the snot coming out of your nose to the fact that you get better, right? Generally, you get the idea. Bad guy, good guy, bad guy, get sick, recover. Okay, that's the immune system. And it's these cells that do it. Now, there's many pathogens out there, and to protect yourself from the many pathogens, you need many cells. Why? Because while these cells have many receptors on the surface, each cell only has receptors of one type. Okay, so it has a limited range of what it can recognize. So to recognize many bad guys, you need many of these cells. Clear? And that's why we're talking about diversity, and that's why the question is, how much of these cells do you need? Okay, so an important maybe thing is, so one cell equals one type of receptor. All right. Now, and so then we need many cells. And these many cells are what we call a repertoire. Repertoire just being a word for ensemble of cells. Everybody happy so far? Good. Okay. So let me draw another line. Okay. So that's why the question, how big, and also what does the question big mean, which I will try to address. How do we measure big when we have complicated things? But first of all, let's start with so hopefully now I've answered this. We're good. What is an immune repertoire? Okay, so now let's figure out, to get to the big question, what are the constraints? Constraints. Because as you can imagine, if I want something to be big and I don't have any constraints, it can be infinite. And that's obviously not the question, because you can come up with one simple constraint, the size of the universe, right? So we're not going to go there. All right, so constraint number one, and we're going to spend a lot of time on this, is you don't want your immune system to recognize your self-proteins. Okay? Everybody agrees with that? You don't want this thing that, kills cells and kills bad guys to kill your own cells. So this is a very important constraint, and we're going to try and understand exactly what that means. Okay, and this, what I'm going to talk about here, is an idea following the work of Rob de Boer and Alan Perelson from 1993 in Proceedings of the Royal Academy of Science, which has been modernized by Grégoire Altan-Bonnet, Thierry Morin, and myself in a review around 2022. I think it's Physics Reviews. So if you want to read more about it, or something's not clear, you can read there. All right, so we're going to talk about B cells for now, because B cells are easier. Why are they easier? So B cell, I'll just draw one of these receptors. B cells interact directly with the whole foreign protein. T cells, as I'll show you in a second, interact. It's more complicated. They get presented bits of peptides. But this is a protein. And there's this. Take some of these over here. And proteins have what are in immunology called epitopes, which are basically just different parts of it that get presented to this cell. Okay, for all practical purposes, it just means bit of protein. And so it interacts with this directly, and right. And when it does, then when it recognizes this is a foreign protein, it generates an immune response, right? So when this, when it recognizes, this means that there's complicated pathways that get activated here. So basically a mess like we just heard about, right? Similar things happen inside. I won't talk about it, but just so you know, and out of that, what happens is that when it gets activated, this cell starts secreting what are called antibodies, which are basically soluble form of this same receptor. Okay, so then you get tons and tons of these. And so then you have, anybody seen this cartoon as a kid, Once Upon a Time There Was Life? Yeah? I can only say it in 3 different languages, but for those of you that know what it is, right? So these are the guys in the, that go around and then okay, right. So now we have an army. Okay, so they're going and they're gonna just basically, what the immune word is smother. They're gonna stick onto different cells, and then that's gonna mark the cell for degradation, and kill it. And at the end of the day, these are relatively short-lived, but these are not. So at the end of the day, if these have been successful, these are the actual B cells. Then they'll go and some part of them will be put aside and given like a flag saying you've been to war, and these will become memory B cells. So next time you see the same bad guy, they'll be even faster doing this. Okay? Yeah. So there's different ways. One of them is they just stick to it, and then there's the neutrophil that we heard about. A neutrophil is like a huge garbage collector, comes along and eats it up. Yeah. So that's, a neutrophil is part of the innate immune system that I was hoping never to mention. But yeah, there's basically this so that's one thing they do. The other thing they do is they attach to it, and then that means that a T cell can come along, and it's this special kind of T cells called killer T cells, which also kill the cell. Sometimes they just basically, just by attaching to it, the organism will find it weird, and again, there are all these garbage removal mechanisms, but this is basically a way for marking it for garbage removal. Yeah. The epitope has no purpose. It's a state. It's a thing, if you want. It's a part of a protein. It's just a part of the protein that can interact directly with the immune system. Okay, so if I have a protein of size 100 amino acids, not every bit of it will be exposed to the immune system. And immunologists like to say, they like to ask themselves, are certain things more likely to be exposed than others? And they call them epitopes. Roughly, yeah. Cells. This is outside. But everything is a protein, right? I hope that's what you got away from Caroline's talk, right? I mean, most things are proteins. So like this is a protein. Like a B cell receptor, an antibody is also a protein, technically. So stuff on the surface of cells, I mean, it can be a lipid if you want, right? But there's also different receptors sticking out in proteins. Antibodies also, so yeah. We'll get to here. They're actually the binding to a protein. We'll dig this open later. Okay. Yeah. That's why we need different. So the name of the game is this. Broadly, okay? You want the stick, as you said, to happen. If I have this, let me draw 2 versions of this. versus if I have this one will maybe stick, this one won't stick. Not everything will stick. This is called specificity. Okay? Not everything sticks to everything, so I need one that will stick to the bad guy I currently have. That's why I need many of them. Okay? Does that answer your question? So the answer is not everything sticks. Yes, that's exactly the question we're asking right now. I haven't given you any answers yet. And I may nod at the rate we're going. Yes. No. Neutrophils are like, they're just garbage collectors. You leave out the garbage, they'll collect it. Even if you've left out your most precious thing, they'll collect it. Yeah, because it's stuck, it's got an antibody. It's like, it's really, imagine then it's smothered with these antibodies. Does it decrease the concentration of antibodies in the blood? Yeah. There's tons, we're in the billion range. We're like, maybe not billion range, but like there's really the concentration. Once antibodies get secreted, they're everywhere. Even if they decrease a concentration, you wouldn't notice it. That's like when you get the blood, think of your blood test, right? You're sick, your B cells go through the roof. It's not like, oh, you go from whatever the normal amount is. I don't even know, because since I haven't had a blood test in ages, but like, that it decreases by 10%, it like goes like 1,000-fold over, and so your doctor has no doubt that you're sick. Same thing for COVID, right? It's a different test, but it's that level of range. For, there you're tracking viral particles. When you have it, you stick it in your nose, the thing goes blue. Again, I haven't had a COVID test in a while, so I don't remember what the stick is, but you get the idea. These things are not like, we're not quantum precision devices here for these kinds of tests. Okay, anybody else? I have to give you one more bit of biology before writing an equation, so now I'm afraid. All right. So, why do I have to do that? Oh, okay, this is why I have to do it. So, we have our B cells. Now, sort of to get back to your question, where do B cells come from? B cells are generated in a process that we'll talk about hopefully someday. But self, non-self. I'll give you the textbook version. How do they recognize this foreign guy and not recognize self? So you have a B cell that's just been generated with its receptors, and the first thing that happens to it is something that's called initial selection. For T cells, it's also called thymic selection because it happens in the thymus. Okay, what happens to this cell that's just been generated is it gets asked two questions. Question one, do you bind to anything? Now, it gets asked this question it's like an exam school setup. It gets asked it in a place where you only have your self proteins. Okay? So there's a bunch of self proteins running around, and this thing gets asked, do you bind any self protein? If it doesn't bind anything at all in the least, then it's probably not a well-formed protein, not a well-formed receptor. Okay? So then you die. Now, question number two, and remember, we're in an environment when there's only your own proteins. You get asked, do you bind a lot? Well, to a lot of things. And if you bind to a lot of things and all of them, and remember the self, then that's also bad news, right? Why? Yeah, autoimmunity, right? You bind to your own cells, you get autoimmunity. So you also kill them. So you leave the ones in the middle. You leave the ones that bind a little bit, but not too much. Are you satisfied as a physicist? Good, you shouldn't be, okay? But that's the picture. All right, so we keep the ones in the middle, and those are the ones out here in the periphery. So why I needed to tell you that is because that takes us from our initial repertoire, which I will call A0, to a functional repertoire, which I will call R, and there's some scaling factor, which I will call F, okay? So these are the ones that have survived this initial selection, okay? Is that clear? I produce, I select, this is what I have after this selection. The selection is only for binding self-proteins. Yes? Wait, sorry, who spoke up? Yeah. It selects, let's think about B cells. It affects both. It's usually explained on the example of T cells, because for T cells we know it happens in the thymus. For B cells it depends more on, in both cases depends on the species of the animal. But it mostly happens in the bone marrow. I'm a physicist, I'm gonna like sweep under the rug a lot of, in this case, this, blah, 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 blah. This is somewhat, but since the word thymic selection only refers to T cells, that's why I thought I mentioned it, especially since I'm being recorded. And, but basically, let's just focus on the algorithm, right? And the algorithm is the same for both. Yes? Yeah. Yeah. No, okay. This is really a question. Imagine I have this cell and I try binding it to 10 to the 6 different molecules and it binds to none of them. Okay? The most likely explanation is this receptor didn't even fold correctly, right? It's just defunct. This can happen, and you'll see probably tomorrow why that is actually a very likely scenario to happen. Okay, so that's this thing. Here, then I'm binding to my 10 to the 6. I'm making this number up. Different self-receptors, and if I bind to all 10 to the 6, or even I bind to a big fraction, or a fraction of those, that's bad news for me, because when it goes out of the thyme or wherever it is to the periphery, it'll be doing that all the time, and I'll be getting rashes all the time, or worse, or diabetes or whatever. So I don't want that. So you see the difference? Okay, question in the back? Yeah, no, so the thymus is known to have only self. Okay, I mean, this is a really complex question, but if you've ever had sweetbread, that's how you know. Okay, sweetbread is the thymus of a cow, of a veal actually, and we only eat it from veal because young veal, because they're pure. As the cow gets older, it gets, biology relaxes, and the thymus becomes less pure, and we don't want to be eating the mug that the adult cow has in the sweetbread. I mean, we're like, okay, so generally this happens all throughout your life. You produce T cells and B cells all throughout your life. However, as you get older, you start producing less. for various reasons. One is basically filling up the bag. As a newborn, you don't have a completely empty bag, but in utero there is some production, but not as much as later. So first you fill it up. That you reach very quickly, you saturate. However, kids, large turnover, they're learning, the immune system is learning in the physical sense. You reach puberty, things slow down. Basically, immunologically you stagnate, and that continues for a very long time until you hit immunological old age. And as my friend Deborah Dunn-Waters like to say, that's around 50. From 50, but not to zero. It's never to zero, but from 50 you become wiser but less naive. So that means your memory pool is larger than your naive pool. Okay, interesting dynamics. There's another dynamic, there's another time scale question. Do you have time for all of this? Which I can give a 45-minute talk about, and the answer is no. But I'm giving you, the textbook pictures for now. I'm basically going to sell you stuff and then tell you it's all rubbish. As a physicist. Okay? Yes? Yeah. Not here. Here we're in the beginning. So for simplicity, think you're an infant. I'm just asking, how do I produce this first? This is called, in immunology, this is called the naive repertoire. All that happened to it, was generated, and I would love to tell you about generation. For now, let's assume I just generate these receptors and the cells, and then I do this thymic selection, which I have now just used the letter F, and all I'm saying is that just the fraction of them get kept. Okay? Wow, I made it from my first page of notes. All right. So, okay, so this is the fraction of selected cells. Remember, these are that means that they did not recognize any self, because we're in this setup where we're only dealing with self. So that means that F is 1 to the minus P to the N, where N is my number of self peptides, and P is the recognition probability, basically. Okay, that means that it's that the probability that the randomly chosen receptor recognizes a randomly chosen peptide. Okay, randomly, anything. Yes, good. Yeah. Wait. Yes, but precisely, I want to figure out how many of them get discarded. Yeah. Now I'm going from biology to physics. Same thing written in biology, physics. Right? Okay, good. So now we're done with the self. So now let's move. So this was the self part. And now let us move over to real life. That means where there's pathogens. So now I'm out in the bloodstream. I passed and I ask, what is the probability of escape of a foreign epitope, of a foreign bad guy, okay? And I'm going to call this PE, probability of escape. So that means basically it doesn't get recognized by any receptor. Agreed? And since these numbers p are small, I can write both of these as exponentials. Everybody happy so far? Okay, so if I have this, then from that, I have that r is minus 1 over p logarithm of pe. That's it, right? So this gives me the size of the repertoire. And R0, the pre-selection repertoire, is minus 1e pn log pe. So pre-selection. I'm going to write rep because I'm too tired to write repertoire. And now, so basically I'm done, right? I mean, I've solved my question now. Okay, now I'm going to assume, huge assumption, that evolution has optimized the recognition probability p. Such as minimize R0. Okay? So basically what I'm saying is that somehow evolution has optimized this initial recognition probability of anything by anything such that it'll have to produce the smallest R0 possible. Okay, so that it generates as little initial cells as possible. Yes? It's like one of the first actually conceptual things I'm saying, so nodding would be good. Yes? Yeah. Yeah, this is a bag, this is not hard calculation, right? This is just a back of the envelope. I'm waving my hands with some equations. I could have said it to you in words. Yes. Yeah. Yeah. But this assumption is basically that it'll produce something that binds stuff, but in a way that I don't have to produce loads of things. Okay? So dr dp equals 0. We agree? Right? I'm minimizing R0 with respect to p. So where do I have my R0? I have it here. So the first term gives me p squared minus p n e to the, it's a pn log p. Okay, this equals 0. So that means that p star equals 1 over n. That's our first result. So what does that mean? So this is our optimal recognition probability. P doesn't depend on P, and PE doesn't depend on P, no. All right. No, I already used this. That would be circular. From this, I got this. Right? To find this R, I took the log of this P. This is now a number. Right? Good. All right. You're paying attention more than I did when preparing the notes. All right, so this is our optimal repertoire probability. And just to write out the rest, what it means is that our optimal repertoire is then just R0 F star, which is minus n log PE, and F star is just around 0.37 as a reminder. The selected fraction, right? This is optimal repertoire and optimal selected fraction. Okay, what does all of this mean? So first of all, what this means is that the minimal repertoire scales linearly with the number of self-epitopes. Okay? So that means that the number of self-epitopes determines repertoire size. Does that make sense? I'll give you a hint. Think about me and a mouse. I had a mouse in the apartment last night. So me and the mouse. Do you think we have the same immune repertoire, size of immune repertoire? Yes? No? No, we don't. I have more. I win. In fact, I can produce every receptor the mouse produces. Okay. What about our self proteins? Do you think we have similar amounts of self proteins or different amounts? Actually quite similar, right? The genome sizes of me and the mouse are not that different, globally speaking. So this is one reason why this is probably not true, okay? But it's a cute calculation. And hopefully now everybody understands the difference between the generated repertoire and the final repertoire. But, you know, it's not the end of the day. Now, where do I go? I go here. It's not the only thing we learn from this. I can surely erase this. Okay. The other thing, so this is point 1 that we learn. It's linear in the size of the repertoire. Let's call it point A since I called 1 that. Okay. B, we learned that, well, the recognition probability P goes as 1 over n, so is inversely proportional to the number of self-proteins. So, yeah.
What you said about human and mouse is true for any pair of
Well,
mammals, right? The generated repertoire is
yeah, so it's okay. So actually maybe what I'm going to say now is going to sort of frame this in more, so I have a lot to say to your question. Let me unload it. Okay, so first of all, every jawed vertebrate has an adaptive immune system. Every jawed vertebrate is every fish you're likely to eat, unless you're a medieval king. And from there on the evolutionary tree, right? There's lampreys, they're fish, they don't have jaws. They also have, well, let's talk about that over lunch. Anyway, we all have basically mammals, birds, reptiles, fish, all have this kind of thing. Now, what I'm sort of presenting here is a view of the immune system as a sensory system, right? I'm saying there's this space of pathogens, and you have this space of receptors, and you want to somehow match them. Now, the problem, when we think about sensory systems, say we think about, vision or audition. It's hard enough to quantify the space of possible stimuli. Here we have no idea what the space of possible stimuli, meaning pathogens, is. And in fact, it's worse. You're now being protected against pathogens, SARS-CoV-2 being a brilliant example, that did not exist when you were born. And your immune system manages. Right? So you have this sensory system that is adaptive, but that can sense things on which it wasn't trained. To put it in a sort of, right? And then the question is, if we think about different species, how different is the space, is this dual space to our receptor, basically? And we don't know. We have no idea because we don't even know how big R is. But given that the mouse and I and probably the lizard, and that's, climbing the wall, and everybody else lives in a quite similar environment, they're not that different. Now, as we know, some viruses come from us from animals, like birds and pigs and so on, and they actually live well in those, will live well. They infect those animals, but they don't infect us until there's a mutation that happens. So there is that step that complicates things. But in this hand-wavy world of, of what I just did, let's say we roughly live in the same fish tank, right? Okay, anybody? Yes? Yeah. Yes. This is very naive. Incredibly naive. It's not going to be terribly wrong in some things, but I already showed you in some ways it is very wrong. Yes. No. No, they just die. Mm-hmm. You think it's wasteful, don't you? Well, that's why we said we want to minimize R0. But, okay, life is wasteful. I think this is right? I mean, whoa. Okay, there is a little caveat. Some of these cells that would fail point 2, they are actually kept as what's called, in the T cell case, as regulatory T cells. And these are cells that can recognize self. And the current view of regulatory T cells is that they basically, if you go over with your immune response, they quench it. So they're like a negative feedback thing. But this is like, this is later on in what I want to tell you. Okay, but basically they get discarded. And if you think this is wasteful, when I tell you about how they're made, you'll be shocked. Okay, it's super wasteful. Yeah. It's as general as the assumptions I made and derived. I don't know, but I wouldn't say it's worth worrying about. I mean, okay, I could have just drawn cartoons for you. I thought as physicists, this is something that speaks a little bit more to you, right? It's really a bag of the envelope argument. That's all it is. I'll hopefully get to the more like solid stuff later. But just to give you, I mean, at least you're following me, at least you're listening to me, so I guess I achieved some goal. All right, so now we have this. And so what this helps us do is actually think about the problem as a covering problem. So this is an idea of shape space, which was put forward by Alan Perelson in the 80s. And it's just it's a phenomenological space. So we say that both receptors and pathogens live in this space, and receptors are balls of a given size. Okay, so there's a receptor here, and they recognize everything that's within this space. And then there's pathogens. Then there's self proteins. So you don't want the self proteins to be recognized. So these are self. And then there's pathogens, and you want the pathogens to be recognized. Right? Pathogens can be anywhere. So you want to cover the space of possible pathogens with these balls. And in such a way that you don't hit the self, but you hit all the pathogens. And basically the volume of each ball is P. Okay? The question is, how many of these balls do we need? And the answer is that, okay, and this P, according to this, should be inversely proportional to number of self. Let's do it this way. Number of self, right? That's what this scaling tells us. And the number, so number of balls, which is R, should scale with N. Okay? This is part of this calculation, but this picture is very useful. Okay? It's basically, it frames it as a covering problem. Right? Packing. And then, if you want, I can now draw balls of different sizes here. Right? And that would be a distribution of P's. Yes? Mm-hmm. Yeah, no, so it's been worked out how long. So one of the other questions is, how long does a receptor have to be? Or how long does the peptide that TCR that recognizes have to be to differentiate basically vertebrates from viruses from microbes? And that gives you a scale that roughly is okay. But in general, it turns out that if you look at the statistics in more detail, the bacterial proteins don't have that different statistics from our own in rough scales. So, but people have thought about this, but it hasn't really led to super conclusive answers. Okay, I just wanted to introduce this picture because it's useful, and we'll be coming back to it. Now, there's maybe smoking gun of this. So now compared to experiments. And this is, of course, hard because we don't, as I said, we don't really know anything. But people have tried to measure things, and this is what they come up with. P is around 10 to the minus 5. So that's the recognition probability. And N, the number of self-peptides just based on size of the human genome, is N. So that means that P is roughly 1 over N. And so that does seem to be at least not inconsistent with experiments. The other thing we had was this fraction of selected, which we got to be 37%. So if we compare that to probability of cells passing what's called negative selection, which is this initial thymus thing. This is 25 to 45%. Okay, there's a citation from this from Hedda Wademan, 2003 in Science. So that seems to be consistent too. Okay. So now we're getting to the biggie, repertoire size. Okay. So we have that R star goes as 10 to the 5th, because now we can put a number in. Where was our repertoire size? N log NP, right? Log 1 over PE. Okay. So the question is, what is that? What is PE? And the answer is nobody knows. Remember what PE was? PE was the probability of escape of a foreign epitope. So it's an effective number. It's actually what you see. We know that it's very, it's probably very small. Otherwise, we'd be getting sick all the time. So we don't know what that is. But we do have some idea about what R star is from data. And, well, it differs, but it's between 10 to the 8th to 10 to the 10th. This is the number of unique cells. You have of the order of 10 to the 11th B cells and T cells separately total, but they come in clones. One cell exists in many, many copies. But unique cells, this is roughly the number. So basically, if then I work backwards, that tells me that PE is very, very small. Okay, you can work it out yourself. And if we were to take I don't think I need this anymore. Okay. So if we assume, just for argument's sake, that 99% of, let me call them pathogens for now, each present 10 epitopes, and are recognized by at least one antibody. That gives me PE to the 10 of 10 to the minus 2, which would give me an R star of 10 to the 4, 10 to the 5, which is too small. Okay, so basically what I mean by here doesn't matter. Let's just call it a reasonable assumption. Okay, so here we fail. And we kind of failed here too. So some of this makes sense, some of it doesn't make sense. But again, it's a way of thinking about the system. And it shows that this problem of basically avoiding self is not negligible. All right. Any questions about self, non-self? Yes? Yeah, these are pathogens. These are self. I don't know, because I mean it depends what you mean by pathogen. If you know, it's how many pathogens you encounter in your life, or now. You know, in your life probably is what you want to do. It's not the total. So there's on the pathogen side, there's a huge sampling question, right? How do you sample the space? But yeah, I mean, maybe there are more. I'll get actually. I'm happy to draw many more of the. I mean, I'm just lazy, right? I mean, some of them you don't hit. I'll get back to. I want to talk about what the ball means now in more detail. Yes. Here. Okay, so the prediction here is clear that what sets the repertoire size is the number of self proteins. So if you have more self, you should have a bigger repertoire. This is roughly true if we compare, and okay, more precisely linearly more. Now we do have a bigger repertoire than mice, and we have a bigger repertoire than fish. And mice have a bigger repertoire than fish. There is a growth of the repertoire. So, but then if we think about it, do we have more self proteins than mice or fish? Well, maybe than fish, but than mice, not clear, because our genome sizes are globally similar. It's not I'm waving my hands. Is this true? Maybe, if we did precise measurements and compared ourselves to, other humanoids, and I don't know. Nobody's done that. Honestly, it's a really interesting question to ask this question across species. There's very little work on that. Yes. I have to tell you what cross-reactivity is to answer that question, and I will in a second. Yes. Yeah. Everything, both. You have a space. I drew it in 2D because I can't draw it in any other dimension. But the idea is that everything, pathogens, and your receptors live in the same space. And the thing I forgot to say, the closer they are, the more likely they are to recognize each other. Yeah. Well, no, because I mean, it's because I can't draw, right? I mean, yeah, no, okay. In this, not in the shapespace picture. In the shapespace picture, I can have different sizes of balls. In this, yes, because I made the assumption that everybody has criticized that I only have one P. Okay? Which I don't think is that bad of an assumption in the end, at the end of the day. But it's fair point. But if you think about it, just think about, that's why I try to draw the balls of different sizes. If it I mean, they could also have different shapes. Or they shouldn't too, because then I'm getting rid of my simplification. In high dimension, everything's a ball, right? So okay. Question nobody asked, what is the dimension of this thing? Nobody knows. Okay, but there is probably an answer to that question, but we just don't know. Okay, but now I have this, I have this, yeah, came out close to that, but you get the idea. That's what I want to talk about now. Okay. Yes. Where are you? Yeah. Yeah. I made that assumption, yes. But probably yes, because the P is just molecular binding. Yeah. 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 Okay. Let me erase something to move on. me actually erase this also, just so that it's not so important. Chalk. Okay, so that was point number one. Remember, point number one was don't recognize self. Point number two is basically match or recognize the bad guys, right? That's why we're doing all of this. Okay, so let's do a fun aside first. And so now I'll use this as an opportunity to tell you a little bit about T cells. So I'm going to draw my cell again, but this time it's a T cell. The B cell has gone. It has this receptor on it, just like the B cell. And now what the T cell does is it doesn't directly interact with a cell or protein. It interacts with a whole machinery. Color chalk. This is called an antigen-presenting cell. It's also part of this innate system that I didn't want to tell you about. It's a dendrite cell. But basically, any kind of your cell is an antigen-presenting cell. Any cell can present stuff. Antigen is a word that I should introduce. Antigen means bit of protein too. A bit like epitope. Technically, antigen are made of epitopes. Epitopes are small, doesn't matter. Anyway, it's for all purposes bit of pathogen protein. Okay. What this cell does is it eats up stuff that's going around in your body and chews it up into short peptide fragments. And all of your cells do this. And then presents a short peptide fragment that's between, well, 9 and, I don't know, 15 amino acids or something. Actually, probably less, 9 and 12 amino acids on this machinery it has on its self-surface. And it presents it, and then a T cell comes along with its receptor. And I drew it wrong because the T cell actually interacts with this scaffold and the protein. Okay? Yeah, 9 to 12, T cell receptor. Okay. Do you want more biology words, or can we leave it at that? For those that are interested, this is called an MHC, which stands for Major Histocompatibility Complex. And the reason you may have heard of it is because it's encoded by genes that are called HLAs, which stands for human leukocyte antigen gene. And those are one of the most diverse genes in our body. And if you're unlucky enough to get a transplant or lucky enough to give a transplant, that's the things that have to match on top of your blood groups, right? It's these things. Otherwise, the person who's getting the transplant from you will think this is a pathogen. You want me to write it down?
No, what's the function of this machinery, as you call it?
Because maybe so basically what it is, it's like, what is the function? The same function as radars on the motorway. Basically, this is a system that eats stuff up and presents it to T cells, and T cells have to it eats up all molecules, also your own molecules. And basically it's basically law enforcement, right? It's making sure that there's nothing in your body that is foreign to you by randomly sampling everything that's in your body. Yes? Yeah. Then a digital system? Sorry, you said digital? Yeah. 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 I mean, maybe we'll get to that. You're basically asking, if this recognizes it as foreign, is that enough to trigger a response? Or do you need to integrate information in some other way? The answer is, one yes is not enough to. I mean, there's a time scale here. Basically, 4 seconds of binding is enough for a T cell to sort of be certain. But how do we get to these 4 T cells? There's beautiful work by Grégoire Tan-Bonnet. The answer is adaptive kinetic proofreading. I'll mention this in passing. But yeah, it's okay. All right. We know that only around 5% of peptides, so you have this 6 MHC, each one of you has 6 MHCs, they're all different, I guarantee you. I mean, unless you're actually a good match for somebody organ donor, you're not gonna match your MHCs. And any of these 6 MHCs present about 5% of peptides. Okay, this is a fact. Just back to the sampling questions. Yeah, MHC is this thing that I didn't want to tell you about that's encoded by this HLA thing. Okay, MHC, since doesn't matter, major histocompatibility complex. Yes, I wrote it. All right. And HLA doesn't matter. This is the gene that codes it. It's just the thing that if any of you have heard of it, you may have heard of HLA. Okay, so let's take a peptide of length 12. 5% is this. Let me take a random peptide, and before all of you protein people shout that no, that this is wrong, forget about it, I'm again waving my hands. 20 amino acid, peptide of length 12, that gives me 2 times 10 to the 14 possible antigens that can be presented. So, if the way the repertoire works is 1 T cell versus 1 antigen. So basically 1 T cell recognizes 1 antigen. That means I need 10 to the 14 different T cells. Okay, in humans, and nobody has sequenced the killer whale before you ask, or some, whale or elephant or whatever, so I don't know the answer to that. But we have about 10 to the 9th different T cells. Okay? 10 to the 11th, 10 to the 12th total. So that means we need, 5 orders of magnitude between what we need here and what we have. In mice, we have about 10 to the 6 unique. 10 to the 8th total. Okay? It's not exactly 12, but it's very close to 12. Okay. Anyway, let's say humans, maybe I could fudge it somehow. Mice are doomed. And so I'll say that this is a fun example before you raise your arms. Okay? All right, so what does this mean? Let's think about the mouse. So if a mouse has that, it's, well, a mouse repertoire occupies about 1 ml of blood. Let's even give the mouse a boost and say it's 10 to the 9th. Well, actually, so if I need 10 to the 14th, that means I need this 1 ml, sorry, it's 1 ml, times at least 10 to the 5. This is assuming that this is around 10 to the 9th. Okay, that 10 to the 9th T cells take up 1 ml. So 1 ml is also roughly the volume of a mouse. So that means I need around 10 to the 5 mouse volumes. Okay, so that means if my friend is here and I draw a box, that this mouse would need a repertoire that is 70 times the size of this box, which is drawn to scale for the mouse. Okay? Does not fit. Now, what's the main assumption we made here? This. I mean, we made many assumptions that, all of you brought up. But let's say the other ones I could make, okay, I'll get 65 and not 70. You get the idea, right? This is the biggie. And this is so not true. And what happens instead is that there's this phenomena which we call cross-reactivity. Okay? Cross-reactivity means that in my shape space, when I draw a receptor, I draw a ball around it, and that's what my ball means, and everything within this ball will be recognized by this receptor. So if now, to use my old color code, if I have a pathogen here, or here, or here, they'll get recognized. Maybe not as well, but they'll get recognized. If I have one here, it won't be recognized. If you don't like balls, think about the Gaussian. Okay? So if this is again my shape space. This is where my receptor is. And this is my probability. I called it P before. So probability of this receptor recognizing the antigens that are here. And they'll recognize with a Gaussian probability older. Okay? The fact that I'm drawing a Gaussian is because I'm a physicist, and whenever I'll do a calculation, I'll make it into a Gaussian. Nobody claims it's a Gaussian. Nobody claims it's a ball, but it must be something like that. Basically, okay, so how do you measure it? You take a receptor, you take different pathogens. Okay, that's actually not how that's how people are measuring it right now. You take different antigens, different mutants, and ask how many mutations do you go away to stop getting recognition. That number over there was measured differently. It's an effective number for, I don't remember how they did the experiment. I can look it up. But it's an effective number for different pathogens, probably in a monoclonal, in a mouse that only has one type of receptor, and whether it lives or dies kind of thing. Very effective measure. Okay. Okay, so now we have cross-reactivity. And so and I need to match pathogens, which is point number 2, which I will write in the following way, with this very complicated equation. So if n is my number of antigens, k is the number of receptors that bind the n antigens, or basically the number of receptors, well, they're there so they can bind the antigens, or maybe number of receptors that can bind these antigen. Okay. This is the number of receptors total. And L is the number of antigens. These are receptors, number of antigens they can recognize. Okay, all I wrote down is a matching condition. Basically, if I have R receptors that can bind, each one of them can bind L antigens, and I have N antigens, each one of them can be recognized by K receptors. I want that to be true, right? So, because the other side of this is that each each antigen can now also, I'm going to draw over myself, can be recognized also by many receptors. Agreed? So cross-reactivity has two sides. Each receptor recognizes many pathogens, and each pathogen can be recognized by many receptors. So then P, my alt P, that a random antigen and a random receptor bind, is just L over N, which is equal to K over R. Okay. I think this is an okay place to stop, and I can just continue after lunch. Now, so may I take these 5 minutes to take more questions, but also, yes, go ahead. No, it's whales, elephants. Elephants don't, yeah. No, that's a P53 mutation. So most cancers have this molecule P53 is a molecule that's involved in many cancers. Basically they have a mutation that saves them, and nobody knows what the downside of that is, whether there's a trade-off with something else. I mean, there's a simple practical problem of zoology of whales, and keeping whales. I mean, whales are not very useful lab animals because they don't reproduce quickly, but no, but I don't think the reason the whale doesn't get cancer is because it has the right antibody. It's because basically P53 is a molecule that gets overexpressed. Cancer is this growth of cells, and P53 is involved in a pathway for this growth, and this is basically nothing to do with the immune system. It's just the whale will not. I mean, I'm oversimplifying, but the whale will not generate tumor or a malignant tumor. Yeah, yeah, no, but those are not the whale. There's another. No, I'm now mixing up. It is, and people are doing that. So now there is basically a lot of this is so okay, this is a huge area of research, and a lot of it is done on T cells. So there's, I mean, you don't need to go to whales. There's people, so pancreatic cancer, for example, is a cancer that is very bad. However, I mean, it's basically a death sentence, but some very small percentage of people are long-term pancreatic cancer survivors. Well, long-term means like 7 years at least, but it's, it's ages compared to the majority of people. And there is a question now about, on one hand, understanding what is so special about their immune system, and on the other hand, the specific vaccines. So what you do is you take the neoantigen, because the thing with cancers is that you produce your own pathogenic molecule. Like every person who gets cancer, there's as always, there's exceptions from this case, but most cancers are a mutation that only happened in you, so you then need an antibody that is specific against your own mutation. So they find that neoantigen, the rogue cell, they develop antibodies against it, and they put it back into you. This is like, I mean, it's beyond trials. Some of this has been, but this is like really the last year. And BioNTech, the COVID vaccine people are really behind. So basically, having developed good mRNA vaccines, the technology was similar to what is needed there. Okay. Yes. Yeah. Well, we didn't consider anything there, right? But okay, microbiome comes in later. So if you want, so far I did R equals F equals R0. What happens next is really there's an R tilde with an F tilde of R, which means that now I'm in the periphery. This is my naive repertoire. And now I meet pathogens, and some of these T cells or B cells will recognize them and grow, and there's population dynamics, and this is, the after, which will be called the memory repertoire in practice, okay? And so microbiome comes here. Yes? No, no. That's why I said that I'm waving my hands, but for the purpose, there's more detail. I did this because I didn't want to explain. People did actually the number 70 they got with more detailed analysis of databases and so on. Honestly, whether it's 70 or 20, who cares, right? The picture's fun, that's why I drew it. But the argument is true. The general conceptual argument, you can do a better estimate of what this is, and it comes out to roughly something similar if you take into structural constraints and what's actually a protein and so on. The argument doesn't change, but yeah.
Very quick question. So if I look at the scale of a pathogen, to me it also looks like an encounter rate problem. So has it been approached in that framework of calculating an encounter rate kernel?
Yeah. What do I have to say about the encounter rate? You mean which encounter rate? The fact that you encounter a virus while sitting in this room with all of us, or the fact that then this virus encounters the right T cell?
Yeah, the virus encounters the right T cell, but there we start to get into different aspects like how large the virus's shape comes into picture and their zone of.
Okay, so when the virus enters you, it basically it grows exponentially. That's what viruses do. Very quickly it's gonna, okay. There's also honing mechanisms. While any of these cells, this is also why they're useful, as I said, sort of, security scanning. They scan your organism. Once they pick up something that they do think in some way is weird, or get a mini signal, they hone it to your lymph nodes. That's why you start getting, your lymph nodes, tonsils, you have stuff here, you have stuff gland, and okay. What I also didn't mention is there's tons of signaling molecules. They're called cytokines. Anytime anything goes wrong, look at the cartoon for kids. It's all there. There's all this signaling between the guys with the, it's all very well explained. It's a mess. I'm not mentioning it. But cytokine storm, again, COVID experience. Remember, people got cytokine storms and they died from over activation of the immune system. It's an amazing system. It's amazing it works, but things can go wrong. But yeah, there's a lot of signaling, honing, there's diffusion. I hadn't even planned to talk about it. Now, so about my plan. What I had planned to do is tell you about diversity today, which I haven't done. But that's okay. Now, in the afternoon, tell you a little bit about optimal immune systems, and tomorrow about traveling waves in the context of viral immune systems. What I propose now is just we continue with this, and wherever I get. Does that sound happy to anyone? Sorry, I can't even talk anymore. Good to everyone? Yes? Yes? Okay, good.